Hello students, today we can see the topic photosynthesis in higher plants. All animals, including human beings, depend on plants for their food, isn't it? Why we depend on plants for food? Because we cannot synthesize our own food, so we have to depend on the plants for food. Now, why we are depending on plants for food? Because plants are autotrophs. What is autotrophs? Auto means self, isn't it? That means by themselves they can prepare their own food. So what food is required for them, that can be synthesized by themselves. So they are said to be as autotrophs. Okay. Now how they prepare their own food? It is by the process of photosynthesis. Photo means light, synthesis of food in the presence of sunlight. Now what is photosynthesis? It is the process by which the green plants prepare their food by utilizing carbon dioxide and water as raw materials in the presence of chlorophyll and sunlight. Okay, the process of synthesis of food by the green plants by utilizing carbon dioxide and water as raw materials in the presence of chlorophyll and sunlight. And as a result of photosynthesis, what is produced? Food is synthesized as well as oxygen gas is also released. Okay. So we can say photosynthesis is important due to two reasons. What are the two reasons? One is it is the primary source of all food on earth. Okay. Then the second one is it releases oxygen into the atmosphere. As a result of photosynthesis, oxygen is released into the atmosphere. Okay. So these are the two reasons for which the photosynthesis is very important. Okay. So you have studied in lower classes various experiments to show that chlorophyll, light and carbon dioxide are essential for photosynthesis. So one such experiment we can see to show that chlorophyll is necessary for photosynthesis. So for this we can take a plant with the variegated leaf. Okay, variegated leaf means leaf with different colors. In 10th class you have studied, isn't it? So take a potted plant with variegated leaf, keep it in dark room for about 3 days. So what will happen? All the starch which is already present in the leaf will be used up. Okay, now keep this plant in sunlight for about 6 hours. What will happen? The leaf again begins to synthesize its own food by photosynthesis. Okay, now we want to show that chlorophyll is essential for photosynthesis. So after 6 hours we have to pluck one leaf and then we have to trace the green colored part of the leaf. So here in the first leaf you can see a part of the leaf is green in color and the border you can see it is pale yellow in color. Okay, so like that we have to trace the green colored part of the leaf on a paper to know that where the green color was there. Okay, the green colored part will be having water, the chlorophyll pigment, the green colored chlorophyll pigment. Okay. Now this leaf is taken, one of such leaf is taken, and after taking the trace we are putting it in boiling water so all the tissues will be fixed. Okay, after that we are putting in boiling alcohol. Directly we cannot boil alcohol, we have to take alcohol in a beaker and then keep this beaker containing alcohol in another vessel containing water, and that vessel containing water should be boiled so that when the water boils the alcohol will also boil. Okay, and why we have to put the leaf in alcohol? So that all the color will be removed, so the leaf becomes colorless, so easily we can differentiate the iodine color in it. Okay, yes. Now after boiling it in alcohol, we have to remove the leaf, we have to wash it and then add iodine to it. So when we add iodine, you can see that in the second leaf you see wherever the green color was there, you can see it has turned into blue color. In this it is slight purple color given, but actually when iodine reacts with the starch it gives blue or blue black color. Okay, so the only the green colored part will be turning into blue color. Why? Because that part only what has begun, the photosynthesis or synthesis of starch has occurred. Okay, so this is a very simple experiment to show that chlorophyll is essential for photosynthesis. This is another experiment to show that light is essential for photosynthesis. So here we have to take a potted plant, and any plant we can take it, and then we have to keep it in dark room for 3 days. Again, for what we have to keep it in dark room? For the removal of the starch. Okay, so all the starch present in the leaf will be used up by the plant. Now after that we have to take a black paper, and in one of the leaf, in a part of that leaf, we have to cover it with the black paper as shown in this picture. Okay, and then we have to keep it in the sunlight for about 6 hours. So what will happen? The part of the plant which is exposed to the light will begin to photosynthesize. Then after 6 hours we can pluck out that leaf where a part is covered with black paper. See, when we cover it with black paper, what will happen? The light cannot penetrate, isn't it? The light will not be entering into it. So that part will not receive the light, and the other part which is exposed to the light will be receiving the sunlight. Okay, now we are removing that leaf, and again we have to put it in boiling water. So all the tissues will be fixed. Then we have to put it in boiling alcohol, so the color of the leaf will be removed. Okay, now we have to wash it and then add iodine. So when you add iodine, you can see that the part of the leaf which is not covered will be turning into blue color, and the part which is covered with the black paper will not turn into blue color. So this is a very simple experiment to show that light is essential for photosynthesis. Okay. Now we can see another experiment to show that carbon dioxide is essential for photosynthesis. Here also we have to take a potted plant and keep it in dark room for about 3 days, so that all the starch will be used up by the plant. Okay. Now take the plant and dip half of a leaf of that plant into a test tube or into a beaker, as shown here, a bottle. Okay, and inside that bottle or inside that test tube we have to take potassium hydroxide, and then cover the mouth of that bottle or that test tube, so that no air will be entering inside. Okay, so inside we have taken potassium hydroxide. In a cotton we can dip it and put it, or we can take a pellet inside the bottle or inside the test tube. Okay, and the mouth should be clearly covered, so that no more air will be entering inside. Now this setup should be kept in the sunlight for about 6 hours, and after 6 hours we have to take that leaf and again put it in boiling water to fix it, and then put it in boiling alcohol, so that the color will be removed. Then add a iodine solution to it, and we can see that what change can be observed. The part which was kept inside the test tube or inside that bottle will not show blue color, and the part which was outside that will be showing blue color. Why? Because carbon dioxide is essential for photosynthesis. And what is the property of this or function of this potassium hydroxide? It can absorb carbon dioxide. So already existing carbon dioxide inside that bottle or inside that test tube will be absorbed by the potassium hydroxide which is kept inside. So no more carbon dioxide is there for the part of the leaf which is kept inside to perform photosynthesis. So that part will not synthesize the food, and the part which was exposed will be utilizing the carbon dioxide, and it will be performing photosynthesis. So the part which was outside the test tube will be showing blue color, and the part which was kept inside the test tube will not show blue color, which shows that carbon dioxide is essential for photosynthesis. Okay. Now we can see some of the early experiments which were done related to photosynthesis by different scientists. One such experiment was done by Joseph Priestley. So he did a series of experiments to reveal the use of or the role of air in the growth of plants. Okay, and later Priestley discovered oxygen in the year 1774. Okay, so Joseph Priestley in 1770 did a series of experiments. One such experiment was he kept a burning candle inside the bell jar. What the picture what you are seeing is a bell jar. Inside that he kept a burning candle, and he found that soon the candle got extinguished. It was not able to burn further. Same way, he kept a mouse also inside that bell jar along with the burning candle, and he found that the candle also extinguished and the mice also died. Okay, and he concluded that the burning candle or the animal which was breathing the air was damaging the air. That means it was using a part of the air for breathing and burning, and they were releasing some other gases, okay, or some other part of the air which was damaging that whole air. As a result, the candle got extinguished and the animal was not able to survive. Okay, then what he did, he took a mint plant and kept that also along with the burning candle and a live mouse. Okay, within the bell jar he kept the candle, burning candle, live mouse as well as a mint plant. And he found that when the mint plant was kept, the candle was burning continuously. Okay, and the mouse was also staying alive. It did not die. So he concluded from this that the plants can restore the air which was used up by the burning candle and the breathing animal. Okay, so the breathing mouse and the burning candle are using a part of the air, isn't it? Some gases present in the air were used up, and that air was released by the plant. As a result, the mouse was alive, okay, and the candle was burning. So in photosynthesis, in relation to photosynthesis, what we can say, the plant was preparing its food. As a result, it was releasing the oxygen. How it was preparing the food? By using the gas which was released by the burning candle and the mouse, isn't it? Breathing mouse. So what they released? Carbon dioxide. So the carbon dioxide was used by the green plants, and as a result they were releasing the oxygen. So when oxygen was continuously released, the animal was using it, the candle was using it, so the candle was burning continuously and the animal was alive. Okay, so this was the conclusion of Joseph Priestley's experiment. Next we can see the experiment done by Jan Ingenhouse. He used the similar setup as the one used by Priestley, and he placed this setup once in dark and once in sunlight. And he showed that sunlight is essential to the plant process that purifies the air fouled by burning candles or breathing animals. Another experiment was also done by Ingenhouse, and he did the experiment with an aquatic plant and showed that when the plant was kept in bright sunlight, small bubbles were formed around the green parts of the plant. But when it was kept in dark, such bubbles were not found. Okay, and he identified that these bubbles are nothing but the gas oxygen, and he showed that it is only the green part of the plant that can release oxygen. Okay, so he concluded that the green parts of the plant can produce oxygen when it is kept in sunlight. Okay. In about 1854, Julius von Sachs provided evidence that glucose is produced when the plants grow, and he found that in the plant cells there are certain green substance and they are located in special bodies. Now we say it as the chlorophyll. The green substance is now said to be as the chlorophyll, and the special bodies where they are present is said to be as the chloroplast. Okay, and he found that the green parts of the plants is the place where the glucose is made, and that glucose is usually stored in the form of starch. Next, we can see the experiment done by T.W. Engelmann. He used the green alga cladophora and he placed it in a suspension of aerobic bacteria. This aerobic bacteria can be used to detect the site of oxygen evolution. Okay, that means where the oxygen is evolving, in that area we can see this bacteria will be accumulating in more amount. Okay, Now he used a prism and he split the light into spectral components, and then he illuminated this green alga cladophora with that split light, and he found that this bacteria was accumulating mainly in the region of blue and the red light of the split spectrum. Okay, so from this what it is clear, that is the plants will be absorbing which light, blue and the red light in more amount, so that they can use it for photosynthesis, and as a result of photosynthesis large amount of oxygen can be released. So he found that the aerobic bacteria were accumulating in the region of the blue and the red light. Okay. In the middle of the 19th century, the key features of photosynthesis were known, and it was found that the plants could use light energy to make carbohydrates from carbon dioxide and water. And the equation to represent the photosynthesis was CO2 plus H2O gives CH2O plus O2. And here the CH2O represent the carbohydrate, that is nothing but glucose, which is a 6-carbon sugar. A milestone contribution to understand photosynthesis was given by a microbiologist, Cornelius van Niel. He was studying on bacteria, that is purple and green bacteria. So based on his study on purple and green bacteria, he demonstrated that photosynthesis is a light dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrate. So the equation can be 2H2A plus CO2 gives 2A plus CH2O plus H2O. Okay, so this is the equation. So here he said that hydrogen from a suitable oxidizable compound can reduce carbon dioxide to carbohydrate. So if you take the first equation, CO2 plus H2O gives CH2O plus O2 here, which is the hydrogen donor, it is the water. So the hydrogen from the water will be reducing carbon dioxide to carbohydrate. Okay, and as a result of that oxygen is evolved. So if you take a green plant, the green plants will be using water as the hydrogen donor. Okay, so the hydrogen from this water will be reducing the carbon dioxide to carbohydrate, and as a result the oxygen will be released out. Okay, now some of the organisms do not release oxygen during photosynthesis. Why? Because they do not use water as the hydrogen donor. Instead of that, they will be using any other hydrogen donor. For example, if you take purple and green sulfur bacteria, they use hydrogen sulfide instead of water as the hydrogen donor. Okay, so here we can say they will be using hydrogen sulfide instead of water as a hydrogen donor. So in such a case, the hydrogen from this hydrogen sulfide will be reducing this carbon dioxide to carbohydrate, and sulfur is there, isn't it? So sulfur will be released instead of oxygen. Okay, so when hydrogen sulfide is used as a hydrogen donor, as a source for hydrogen donor to reduce carbon dioxide to carbohydrate, then oxygen will not be released. Instead of that, sulfur will be released. Okay, so he conferred that, that is, he concluded that oxygen which is evolved during photosynthesis by the process of photosynthesis in green plants comes from water and not from carbon dioxide. Normally we think that carbon dioxide is a gas and oxygen is a gas, so oxygen will be evolved from carbon dioxide. But Cornelius said that it is not from carbon dioxide the oxygen is evolved, but it is evolved from water. Okay, because water is the hydrogen donor from which the hydrogen can reduce carbon dioxide to carbohydrate, and as a result the oxygen will be released out. Okay, and it was later proved by various radioisotopic techniques also. Okay, now the correct equation of photosynthesis is 6CO2 plus 12H2O gives C6H12O6 plus 6H2O plus 6O2. So water will also be released as a product of photosynthesis. Okay, so this is the equation, correct equation for photosynthesis which is followed now. Okay.